Before the final speaker this morning, I want to point out there's a few names here of people who should collect checks or tickets or other important things. So maybe you should check they're not on that list. Okay, thanks. And the third speaker this morning is Ed Marcotte, who will talk about steps towards identifying disease genes.
Well, I was going to, as I noticed the beautiful sun outside, I was actually going to propose we skip my talk and go for a nice romp around the fields. But nonetheless, you're all trapped inside now. So let's get started. I'm going to tell you about some work that we've been doing in trying to predict the consequences of perturbing genes. So how does, if you affect a gene by mutation or knock it out, how does that affect the organism? And is this predictable in any capacity? And before I get started, I want to point out a few names, and in particular, In-Suk Lee, who was a postdoc in my group for several years and has actually just left this past month off to his own faculty position. And we're going to see a lot of work from In-Suk throughout the talk. A few other people play into this that I'll just point out beforehand. In particular, I'm going to introduce the models initially, but then I'm actually going to show you a fair bit of kind of experimental work testing the models, and this will be work of Ziwa Lee and work with a group of Andy Fraser, in particular Ben Lehner and Andy Fraser at the Sanger, and a few other bits that I'll try to introduce as I go along. So, all right. So when the human genome was sequenced, and now many other genomes, there are a number of big questions that came out of this. And you've been hearing about many of these throughout the conference. In particular, one of the sort of obvious ones is that if you have 20,000 genes and you look at the entire history of what we've learned throughout biology, we know the functions of a few thousand of those to any detail. And so what do all these genes do? We don't know. And if it took the history of biology to get functions for a few thousands, how do you accelerate that process and start discovering functions for thousands of genes in a more efficient fashion? One aspect of this is how do they all work together? Can we start to think about wiring diagrams for cells and the associations among the genes, sort of logical associations among the genes? Now there's a related notion to this, it turns out, that another big question is that personal genomes are really here already and are coming. You've heard the 1,000 Genome Project and all these sorts of things. How can we relate the variation that's being discovered in all these genomes to variation in the phenotype, the traits that we inherit, the diseases that we're susceptible to, our drug susceptibilities, these sort of things. And can we start to rationally identify which genes are associated with diseases and traits and so on? So in thinking about this first question, the sort of how can we discover functions for thousands of genes, we've recently been able to make some interesting progress on thinking about the second question. And so I'm going to take you through this process. And it sort of all falls into the same, this general notion that we would like to have a magic model that allows us to say, all right, we've observed mutations, or we knock out a gene, or a drug targets a gene, something like that. We perturb the system. What happens afterwards? All right, so this is the goal that we're sort of looking for. And of course, the rationale, as I've been pointing out, is that if we could do this, this would help us to interpret the variation we see in our genomes. But it's a very difficult task, and right now our ability to do this for any arbitrary gene is essentially negligible. So more importantly, we have a great deal of data now being collected by groups all around the world, and it ought to be possible to at least start developing approaches to take advantage of that data. So we're really sort of thinking about pragmatic solutions that use the current data to start addressing this sort of an issue. And of course, one of the big points is that this will get us to kind of rational directed identification of disease genes if we can do this to any sort of capacity. And one sort of maybe non-obvious aspect of this is that if we can do this, and the models that I'll talk about would actually be very useful in particular for studying traits and diseases that involve many genes participating together. So even possibly massively polygenic cases. And I'll actually make the point that, in fact, the models I'm going to show you do have some strength for these massive polygenic diseases, which are quite difficult by other approaches. Okay, so what's in the black box? Right. So I'm going to talk to you about a class of models in which we consider associations between genes. So we've talked about networks of various flavors so far in the conference. We're going to think about, in particular, a class of networks in which we're thinking about the genes being linked in a graph if they participate in the same process in the cell. And we call these functional gene networks. And we can learn these from many of the kinds of data available to us in biology right now. We've been hearing, for example, about DNA microarrays and so on, protein interactions. Many of themselves sort of naturally lend themselves to learning associations between genes. And the key idea that we're going to exploit, so this is it for the talk, is that if we can learn these sort of networks efficiently, then we can then propagate information through these graphs in interesting ways. So if we know a set of genes, for example, associated with type 2 diabetes, then their network neighbors are going to be more likely to be associated with type 2 diabetes. If we know a few genes that participate in a function in a cell, then their network neighbors are likely to be involved as well, right? That's the key idea. So if you take nothing else from the talk, everything else is going to be sort of application and validation of that notion. Right. By the way, I should say that feel free to interrupt at any time, so that if it's not clear, then it's to you, then it's probably not clear to somebody else as well. So, all right. So the outline of the talk. Let me introduce this notion of functional gene networks to you in a bit more detail in their derivation. And then the way that we've been studying these is we've really been trying to do sort of a hierarchy of models. We test initially in a lower organism, yeast, where we have a lot of data and relatively few genes, and where we can actually do experiments in the lab to test the models in a very rapid fashion. And so by doing this, we've been able to validate most of these notions. Once we're comfortable they're working in yeast, we're sort of moving up the ladder of complexity, so moving up into animals. So then I'll introduce first some validation in yeast and then show you this working in an animal, C. elegans, and moving into other systems, including plants and humans. All right. So let me sort of talk about this notion of these functional association networks. So what we have available to us at this point in biology is really remarkable, that with a number of these technologies like the DNA microarrays and interactions and so on, protein interaction screens and so on, we have a great deal of data available digitally and from labs all around the world, deposited into open databases, open access in many cases. So we have literally millions of observations that we can take advantage of varying quality and varying subject matter and so on, but a great deal of data available to us. So we have all of these sort of various sorts of data available to us and really concentrated on a few organisms in particular, like yeast and worms and mice and humans and so on. We also have the sequence information, so information we learn, say, about one organism, we can often transfer to some extent via the evolutionary conserved components to another organism. And so this is sort of the playground we're in, is that we have all of these various sorts of data, very noisy. We have it for different organisms, and we're going to try to, out of that, distill some sort of sense. And the notion of a functional gene network is that many of these kinds of data we can express, we can reinterpret long after they've been collected for different reasons altogether, as telling us something about associations between genes. So the obvious ones are things like if we've measured that two proteins interact, then we know that there's probably some functional association between the two genes. So gene A and D, their encoded proteins physically interact in the cell. So we can start to build these adjacency matrices that describe pairwise associations among genes. Some of the other kinds of data, for example, DNA microarray, we can infer pairwise associations. So across a bank of microarray experiments, we might have a vector of expression, and we could look at correlations in the expression to get to pairwise associations, and so on. So we have all of these sort of various types of biological data from which we infer pairwise associations about the genes. We have the kind of the sum total of what's known about the processes the genes operate in. So we've heard earlier about the gene ontology annotations. That is essentially our set of known pathways. So we can treat all this in a supervised framework, where the goal then is to basically learn which genes are likely to operate in the same process from all of the evidence available to us about the associations, all of which is noisy, incomplete, and has all sorts of its own particular properties associated with it. So essentially, this is really just illustrating that we're in a supervised framework. We have known associations between the genes. We, in some way, look at the existing data sets, and there's a great deal of work in sort of testing these and in effect, weighting the associations we see from different methods, combining them in some way, And ultimately what we end up with is an adjacency matrix describing pairwise associations between the genes, where we're trying to construct a matrix in which the entries are the framework we're working in is the log likelihood of two genes working in the same biological process. Okay? That, if you threshold it at some level you're comfortable with, is one of these networks. You don't have to threshold it, of course. You can treat the entire set. So this gives us some estimate of associations among genes, and this is some estimate of the wiring diagram of the cell. Okay? All right, so a bit more concretely then, we have, as I say, these different specific kinds of data, genetic interactions, DNA microarrays, and so on. What we've done is to take all of these various kinds of data, and for the yeast network I'll tell you about, this is something like 20 million observations overall of various flavors. And we integrate these using regression models in a Bayesian framework and to learn the network itself. And much of the work is that for each of these kinds of data, we have to go in, because there's such a diverse pile up of data from different groups and so on, they have their own specific noise. We have to sometimes develop data intrinsic scores to say, convert discrete observations like protein this interacts with that into continuous scores to try to account for the quality of the interactions, that sort of thing. So we've had to invent a lot of these scores, noise filters, these sort of things. But all of this goes in to give us a functional network. And at that point, we've synthesized all this together into a fairly easy framework to now go do interesting things with. And so that's essentially why we've broken it into this two-step process, is that having this in place now simplifies a lot of the downstream analyses. So, for example, if we cluster the genes by their connections, then that gives us some semi-objective estimate of what the systems of genes are in a cell. Or if we look at a particular gene that we don't know what it does, if we look at its neighbors in the graph, this gives us a chance, if we know what they do, this gives us some information about what that gene does. And this gives us a sort of a simple vehicle for rapidly getting at least putative functions. And this notion that we're going to be exploring later is that genes associated with a trait, their network neighbors are often going to be associated with the same trait. And this is the one we're going to be exploiting later on. I'll give you just a bit more of a feeling for how we're doing this integration. So we have the known annotations of the genes, which is very incomplete, noisy itself from gene ontology and from other approaches as well, other ontologies. For a particular set of data, so let's take in this case, this is a set of 50 DNA microarray experiments from the following paper in which they're measuring genes expression across a set of conditions. Each of 6,000 yeast genes therefore has a vector of length 50 of its expression pattern. And if we look at something like the correlation coefficient between expression vectors, we can rank all the genes by the correlation coefficients, and in bins of genes, we can calculate the log likelihood ratio for being in the same process versus not being in the same process. And this gives us some sort of a curve so now we have an interpretation for the genes that we happen to measure on this particular experiment. If we see them correlated so far, that gives us a likelihood of so much of being in the same process in the cell. Right. That's just this particular experiment. Of course, we have lots of sets of DNA microarray experiments that we can do this on. For reasons I won't get into, it turns out to work much better if we're analyzing kind of coherent sets of experiments rather than just piling them all up together. But we can now take these sort of models, we can fit these with regression models, and then extend from the annotated set, which is what this is evaluated on, to all the genes that we don't know annotations for. So we're making this assumption that the genes that we don't know any pathways or functional associations for at all will behave the same as the set that we do know some pathway information for, which is perhaps a leap of faith, but it seems, I mean, in practice, it's the only thing we can do. All right, so we fit these with regression models, and then we can integrate these in various ways. And as I say, we're integrating these in a sort of a Bayesian framework where we're taking such models from lots of diverse sets of microarray data, protein interaction data, and so on, each of which have their own particular kinds of scores that we have to play with and develop through doing all of this in a recall precision framework, and then integrating them together until we end up with these models I've been describing about. So much of the work involves going in and looking at the individual data sets, developing scores, and so on. So just to give you a flavor, not all, for example, sets of DNA microarrays are useful for this process, and we spend a lot of time going through and doing these tests. So for example, here again is the correlation between genes expression vectors and testing on this log likelihood ratio of being in the same process in the cell. And here's one that works fairly well. So you see a relationship between the log likelihood and the correlation coefficient. And here's a set that really doesn't work very well at all. And it's not obvious why this set wouldn't work and this set would, but in fact we find many that don't actually, where the co-expression of the genes doesn't actually confer information about the functions of the genes, some of which we can rationalize and some not. Okay. We do, as I say, all of this in a recall precision framework, and we can then start taking, so for example, each of these curves is a different class of data with many individual observations or sets possibly integrated together into it. So coming from DNA microarrays, from comparative genomics information, where we're looking at patterns across different species, from genetic interactions, from measurements of the physical protein interactions in a cell, and all these sorts of various measurements that go into it. And then in the end, we integrate these in some fashion. So this would be sort of an integrated network in this particular case. And this gives us one of these very complex functional networks. So for example, our current model of yeast genes spans about 95% of the yeast genes and has about 100,000 reasonably high confidence edges in the graph. And that's a synthesis of about 20 million various observations. All right. And the precise interpretation of each edge is that each vertex is a gene and each edge carries a weight, and the weight is the log likelihood of the two genes operating in the same process in the cell, okay? Pathway, etc. And there's a lot of structure to these things. We don't actually look at these graphs very much. It's just sort of useful as a sanity check. Instead, we deal with the adjacency matrix itself. But you can see that there's a great deal of structure and that it's very non-homogeneous. And these sort of structured regions correspond to systems in the cell. And so partitioning this graph or clustering the genes by their patterns of connectivity returns us some measure of systems in the cell. And we can go through and see, for example, is this a new system or a known system? It may be that all of these are unannotated, and that would be a new system that we would have discovered in that case. Okay, so what I'm going to do from here on out then is to develop this framework. I'm going to show you how we've applied it to a few different systems. And much of what we've done for the last couple of years now has been to sort of put our money where our mouth is, as it were, and go into the lab and do experiments following up the predictions that come out of these models. And for a long time, we had sort of worked on developing the framework, working, as I say, within recall precision kinds of frameworks and so on, where we could see how we were doing, relatively speaking. We got to a point that we were confident enough to start testing these things, and now we've tried to do all this in an environment where we've picked systems where it's simple to go from a prediction coming out of the networks to rapid kind of experimental tests and try to keep that cycle fairly close as a sanity check. All right. So the way that we're going to do this, we're going to start off, because this is a functional network, the assertion is that these two should be functioning together in the cell. Any linked genes should be functioning together in the cell. So we're just going to try and predict functions for genes. So again, we have a set of genes, we'll call them seed genes, exemplars, that we know have a function of interest. And then if we simply take any other gene in the genome, so any other node in the graph, we can sum its edge weights to those genes, and that corresponds to a naive Bayes estimate for being in the same process. Okay? So we can rank order all the genes in some way, and then that gives us now sort of a list of who you should test in order for being in the same process. All right? So I'm going to take you now through kind of an experimental test of whether that works or not. And just to put this in context, one of the systems that we've now tested in a fair bit of detail is the genes that are involved in manufacturing ribosomes. All right, so ribosomes are the main machineries in cells that make all the proteins that we hear about. And the ribosome itself is very complicated. The machine that makes the ribosome is even more complicated. We already know something like 150 genes involved in this manufacturing assembly line. And within a cell, you have a nucleus and various compartments within the nucleus. And proteins for the ribosome are manufactured by ribosomes in the cell itself, and they're shipped into these compartments, and then there's a whole series of events in which the various pieces of the ribosome are processed and assembled and chopped and so on and shipped through various compartments before they finally make the ribosomes themselves, which are these, as I say, very complex machines. So in yeast, something like what's the 78 proteins involved, very large non-coding RNAs, very complex machines. All right. There's a lot of evidence that says many more genes in this pathway remain to be identified, and so we did exactly what I've described. We take the 150 we know as our seeds. We see who they're connected to in the network, calculating this sort of naive Bayes criterion for being in the same process. We can test the general predictability of this using kind of rock statistics, where we do this in a cross-validated sense on the known set, and they predict back very well, so it says that they cluster in the graph structure. And so here's a particular one that we don't know the function of this gene, but it clusters with a number of other genes, and all of these green ones are known to be involved in this process, so that's a pretty good candidate. And we've now gone and we've taken, we took about the top 200 predicted ones that came out of this test, and for various reasons, we ended up sort of settling on about 100 that we could test in detail for technical reasons. 50 of these were essential. If you delete these genes, the cell dies, and 50 of these were of the flavor that if you delete them, the cell is okay, but if you maybe shift it slightly out of optimal conditions, it gets quite sick. All right? And I'll spare you all the gory experimental details, but we did a couple of years' worth of work summarized in a little corner over here that from graduate student Ziwa Li, who's really done a wonderful job on all this. And we've tested now a whole series of different aspects of their involvement in the biogenesis of ribosomes, and we've been able to validate about 40 out of the 100 turn out to be legitimately in this process. We actually think that many of the remaining ones are too, but that we're sort of missing them in the conditions we're looking at. And we know this because if we inactivate pairs of them, then we actually can implicate them in the process. But there's sort of a higher order dependency we haven't quite explored yet. And out of this ones that we've tested, for various reasons, about 25 of these we would consider to be completely new genes in this pathway. All right, that we now have an idea of where they're working. And just to give you sort of a flavor for the kinds of things that we're finding at a very high level, then, for example, one of the things that we find is within the ribosome itself, there's this very large non-coding RNA, several of them, and the non-coding RNAs are modified chemically at a number of positions by specific enzymes in the cell that presumably helps in the functioning of the ribosome in some way. And one of these is the methyl groups. Chemical methyl groups are attached to various of the nucleotides in the non-coding RNA. And the enzymes that performed this methylation were known for many of the sites, but there was one site in particular that it was never known what the enzyme was, and this was one of the ones that happened to fall out of our analysis, is that we discovered the last unaccounted for methylase out of this, and we could show that very specifically it modified the RNA. And we found now, as I say, about 25 new genes in this process. Okay, so that was sort of a large test of the principle, and it appears to work very well in that test. Here's something that's completely unrelated. It's a second test looking at a completely unrelated system, and this, I hope, will sort of illustrate the power of these networks. Is that they're spanning many different systems, and you would probably do, I guess, better if for any particular system you went in and learned specific classifiers and so on to learn new genes in that system. But these in a sense, have all of this in one model that we can query rapidly for different systems. All right, so this is a completely different case in which we've now done some experimental tests. In this case, looking at in yeast cells, and I don't know if you can see, I'll trace it out. There's a bit of a cell there, and when you add a pheromone to yeast cells, they grow in a polarized direction to find the yeast of the opposite sex so they can mate. And this is a model for many systems, such as, for example, when your neurons grow out to connect to each other, or for many processes in development when cells are growing in a directed fashion. So directed growth. We were looking for proteins that are involved in directed growth, and we could do this under a microscope by labeling any arbitrary protein so that it glowed fluorescently, and we could see whether it went to the tip of the cells or not. And we had done this for many proteins, and we'd found about 40 proteins that went to the tip of these growing cells. And we use these as our exemplars to query the network in the manner that I've described, and took about the top 100 candidates from this approach and tested each of those for this property. And out of the 100 or so that we tested, we got about 37 new candidates that came out of this. So essentially, we doubled the number that we knew using this network approach. And it really gave us a very efficient filter for doing the experiments. The experiments are a pain enough that we didn't want to do everything, but in doing this very focused way, it allowed us to focus in and get some sets out. And you can see all of these are various proteins that now go to the tip that we can now argue are involved in this process. Some of these are completely unknown function, but we now have some notion of where they're going in the cell. Okay, so this all sort of gets at notions of function. You can see in both of these examples, we're not by any means hitting 100% accuracy in the predictions we're making, but we're in a regime where we're getting things that are useful to an experimentalist to go in and discover new information. But where we set out to go was we wanted to be able to look at the perturbation to the system. A slightly different statement. So we'd like to know if I mutate or perturb a gene, what are the consequences to the organism? Can I predict, for example, genes that give rise to something like a tumor or diabetes or lifespan or these sorts of things? We've explored doing this within the same framework. So it's again, we have seed genes, but now they're associated with a trait or a phenotype. And we're again taking for any particular node the sum of the edge weights in a network to the seed genes, and that's this naive Bayes estimate of being in the same process of these. So we're basically assuming that the edges that we've learned, which is formally the log likelihood to share a gene ontology term, is really specifically what we're learning, is going to translate into something meaningful biologically. And we find candidates and then look to see whether they make sense. All right, and we can test this again in yeast fairly well. Because of an interesting phenomenon in the field that's really quite remarkable, where all of a group of yeast biologists went and inactivated each gene in yeast in turn. And since there's 6,000 genes, they created 6,000 strains, each of which has a different single gene inactivated. And these are generally available. You can buy them quite cheap. So in our freezer, we have all 6,000 strains. And for any particular strain, you can just go test it for whatever trait you're interested in. And then that will, if you see that trait apparent in that yeast strain, that tells you that that missing gene is related to that trait. So you can do these sort of studies in a very efficient fashion. And people have now tested this entire set for many different traits. There's at least 100 different traits more that have been tested in this fashion. And we could go evaluate these kind of in a cross-validated sense for if all of the genes that give rise to a particular trait cluster in the network and are predictable back, as I say, in a cross-validated sense using the network. So this is, again, ROC analysis showing the true and false positive rate of the predictions for just sort of a smattering of things. So if we knock out a gene, does the cell live or die? That turns out to be a predictable trait using these models. Does the cell have longer or shorter telomeres, so the bits and the ends of the DNA? Do they germinate more efficiently or less efficiently? Do they take up iron more or less efficiently? So all of these things turn out to be reasonably predictable. And when we look across the sets, we went through the literature and gathered up 100 of these that we could get our hands on, 100 of these tests involving very different numbers of genes and so on, that this is the area under the ROC curve for each of the 100 tests in this, just as I showed you. We can see that, in fact, the phenotypes are quite predictable compared to random gene sets of the same sizes. And this really starts giving the suggestion that we ought to be able to use this for disease gene discovery, right? That's where we're headed. And we could test that a little bit in yeast because there are genes that are conserved between yeast and humans in which failure of those genes in a human causes a disease. These are often diseases of cellular phenomena in some way. And there's about 28 human diseases that have clear yeast orthologs for the genes that are involved. And we could look at those. And again, for those gene sets, we see very good predictability compared to random, in fact, even better than some of the other traits. Just to give you a flavor for what these are, for example, if you mutate any of the genes that are involved in kind of the core energy metabolism of cells, in the glycolysis pathway, this gives rise to hemolytic anemia. And these genes are well connected in the networks, so if you know a few, you can predict the rest by virtue of network connectivity. Other sorts of things. Here's a leukoencephalopathy with vanishing white matter. It turns out to be all of the genes that encode all the proteins that form one physical complex in the cell. Okay. Now, the point of this is we should be able to do new discovery with it, and we've tested this in this framework where we take something that is a very polygenic trait in yeast, which is the genes that determine the shape of the cells. So normal cells are sort of these nice little round oval kinds of cells. Turns out that there's a set of genes that if you knock them out, you get these long elongated kinds of cells. It's very complex. We have no idea why they're doing it. But it turns out we don't need to understand why they're doing it. We can take the ones that are known to give rise to this, use this sort of network ranking scheme, and go test those. So we did this. We had a set that we knew gave rise to this. We tested the sort of the 33 that were most linked to those. If you knock out the genes, what happens? And so 16 out of the 33 tested, in fact, gave us new genes that gave rise to this trait, and that's sort of less than 2% expected. So this is an example of something that's very polygenic, and yet we have predictive capacity even in the absence of an underlying model for why these particular genes change the shape of the cells. But now, since we know which genes are involved, we can try to go back and rationalize that after the fact. Okay, so for the rest of the talk, what I'm going to do is show you how this extends into more complex systems. We could anticipate lots of reasons that, let's say we want to try the same strategy in multicellular animals. We could think of lots of reasons why this would fail. In the end, it was an empirical question, and so we tried it, and remarkably, it seems to work reasonably well. So I'll give you sort of a flavor of a few of the things. I've shown you that, for example, we can use this to predict proteins that change location, involved in making ribosomes, cell shape change. We've tested this now a fair bit in little worms, and I'll give you some examples of predicting genes that increase the lifespan of the worms and that reverse tumors in a very simple model of tumorigenesis in the worms. All right. So to do this, we have to have a network in these organisms, and so we set about constructing one of these functional networks for the animal C. elegans. So it's much more complex than yeast, so it's closer to humans, at least in the gene complexity, so about 20,000 genes, thereabouts. We had a lot of data that specific in C. elegans, and which is one of the reasons that we had chosen it. It also turns out to be very easy to do the follow-up tests where you can inactivate any gene at will in a fairly straightforward way and look at the consequences at an animal level, which is obviously very hard to do in many other systems. So we had, again, the kinds of data where the sorts of physical interactions among the proteins, or where one protein had been purified and other proteins came down with it, and you ask who they are, so looking at complexes. We had a lot of DNA microarray data sets, comparative genomics kinds of approaches, genetic interactions, and so on. And we synthesize all this in essentially the same framework I've described, learning the log likelihood for pairs of genes to participate in the same process in the worm. And our network spans something like about 80% of the genes in the genome, with a little under 400,000 edges in the graph of some reasonable confidence. All right. Now, like in the yeast case, in C. elegans, there has been the ability, as I say, to inactivate any gene, and this is easy enough there's a technique for doing this which is simple enough that you can do this on a genome-wide scale. So you can scan through 16, 20,000 genes and look to see if you knock out these genes, what happens to the animal. And people have now done this measuring for different sorts of traits. And at the time that we did this, there were about 40 such scans had been done. And so we could take, for each trait that was measured, we could take the genes that were affiliated with that trait and ask, again, in this sort of cross-validated rock plot test, whether or not they were predictable back. So if we mask a gene, can we predict it back from its brethren that give rise to that trait? And you can see some of the traits here. So these are, if you knock out these genes, the worm gets smaller, or if you they become blistered on the surface, so there's some defect in the cuticles on the worms, or the lifespan increases, or all sorts of various sorts of things. And what we find is out of the 43, about 29 of them were actually strongly predicted. I'm showing some examples here. 10 of them moderately. There's a few that weren't predicted at all, and we can kind of rationalize why that is. But in general, we had a fair bit of predictive capacity for even these very complex traits. And again, one of these is, does the worm live or die if you knock it out? And what this tells us is that the genes that are essential for the organism are connected preferentially in the network. So it tells us something basic about the essential genes. I'll give you a bit more detail about a few predictions. So one screen that had been done was to find genes that increase lifespan. These are very difficult assays to do. But essentially, you knock out a gene and you measure the lifespan of the worms that have had that gene inactivated. And what you're looking for is for the lifespan to increase, right? It's very easy to decrease the lifespan. That's no problem at all. But to increase the lifespan is tricky. And of course, the object being that, in theory, you could then develop a drug that targets the product of that gene in some way. And then that would possibly be something that would increase lifespan of humans, ultimately. So there were 29 genes that had this property that were found in a large screen. So we could use these as a seed set, find the top-ranked candidates from this worm gene network, and then try to validate these. And actually, at the time that we had submitted this paper, we were very lucky in that another group actually went and tested a number of additional genes, including ones in our candidate gene list that we had made blindly before that. And so out of the top 50 that we had predicted that they tested, 10 of these actually turned out to increase the lifespan of the worms, and about 20 out of 200. So this is sort of in the ballpark of where we're operating with most of our predictions. We're sort of in the 10, 20, 30, best case scenario, 50% accuracy kinds of predictions. But nonetheless, getting in the useful range. And just to give you a single example, this particular gene right here, when knocked out, gave a 50% increase in lifespan. So this is a dramatic increase in lifespan. What was even more interesting is that this gene is essential for life if you knock it out in a young worm. If you knock it out in an adult worm, then they live 50% longer. And it was sort of puzzling to us why this had come up high in the list before, and this rationalized it, is that it only increases the lifespan when it's knocked out at a certain stage in life. So. A second example is that in C. elegans, there's a very simple model of tumors. And if you knock out the genes that are involved in a particular process, in particular this tumor suppressor called retinoblastoma, then you get this sort of bumps down the body of the worm. This is the model for worm tumors. This is the analog in humans. Mutations of this gene lead to a very nasty eye cancer, retinoblastoma. Now, a few genes were known that in a worm that had this tumor model already, there were actually 6 genes that were known that could reverse this. And you think about this in a drug scenario. If you knock out one of these genes, then you'd like to be able to cure the tumors, right? So you'd like a drug targeted against one of these genes. So if we use these 6 genes as our seed, and we find the top candidates, we could then go in and inactivate each of those genes in turn and test in the lab to see whether or not they reverse. And this is with our Ben Lehner in Andy Fraser's lab, went through, tested 170 of these genes, and we actually found 16 new genes that would reverse a tumor in this tumor model. So 15 of these have human equivalents, and so the idea now is to test these in mouse models for tumor to see if they actually retain the capacity in a mammal. We have no idea if it would hold up or not, but at least in a worm model, they're interesting genes that are relevant to the pathways that are coming out of these sorts of things. Okay. Now, what we've tried to do now, that we've done other tests in the C. elegans as well, and it's been sufficient to convince us that the general strategy appears to be working. There's a lot of things that we can obviously improve. So we can certainly improve our statistical models for how we're learning the gene-gene associations. We could certainly improve our models for inference off of these and so on. So they're good enough now, in part because we've spent so much time learning how to parse out the individual data types and to thinking about the scoring for the individual data types, which was actually a lot of the work involved in getting up to this point, sort of thinking about what sorts of systematic noise were present, that sort of thing. So we've taken essentially the methods we've had, and we've started extending these into higher organisms. And so here's an example where we've been applying this to plants now. And so we've gone and INSEC has gone and calculated, using the same framework that we've developed, a network for the genes of the mustard seed plant. All right, so this is a sort of a canonical model for flowering plants. It's a very complex organism, so more complex than a worm. We have a network that spans about 70% of the genes with something like 1 million connections between them. And we can make these similar sorts of predictions. And we've been working with a group at Stanford led by Sue Rhee, who are doing all the hard experimental work to now see whether these approaches will continue to apply in a plant case. And so these tests turn out to be much harder than in the worm case. But what biologists have done, again, is they've inactivated many of the genes of plants and just stored the seeds away without studying them. And so in principle, you can go make a prediction and go get that seed from the seed bank and grow it up and see whether it has the property you're looking at. And so this is an example of, now it turns out to be slightly trickier than that would sound for various reasons, but this is an example of a gene that we had predicted would be regulatory for drought resistance. And when we knock out that gene, here's the normal plant on the left, here's the mutant. You can see the leaves are browning already, these wilt easier, and so on. So this is a validated prediction where we could test hundreds of genes. In the other cases, here we're sort of at the stage of testing 5 genes. So we're already sort of, the technical complexity is such that these are much harder to test. But out of the 5, it looks like so far about 3 out of the 5 are working for the predictions that we have. And this is very preliminary work because there's a great deal of genetics that has to follow up on this to verify that the trait tracks with the mutation as you cross the plants and so on, and make sure that it really is what you think it is. So this one we verified. And given that this is working now, we've actually tried to now extend this into rice to see if we can start getting affecting traits of economic importance like pest resistance and stress. Okay, so let me wrap up with what we're doing now in mammals. So you could argue again that we're even more complex than all of these, and so it should be disproportionately difficult to generate these sorts of models and have any impact on human disease. We have so many different cell and tissue types, and that the data is not well behaved and so on. But I think the reality is that we're probably not that much more complex. And so again, it's an empirical question. So we've gone ahead and started generating these models for human and mouse to see whether or not they work. So we have a similar network for human genes that spans about 16,000 genes. And what we've had to do to get here is we've actually had to transfer information from our previous networks forward. So there's we're borrowing from other systems. There's yeast data propagating into the human network, worm data propagating into the human network, fly, and so on, where these measurements have been made in much more detail, and we can push them forward by orthology in the genes. Similarly, we have a network in mouse, and this was actually kind of an interesting case. There were 9 laboratories that got together in a contest called MouseFunk, led by Fritz Roth and Tim Hughes, in which they masked the identities of all of the mouse genes and masked the known functions of 10% of them and gave them to the 9 laboratories along with a lot of functional genomics data. And the goal was to predict back the gene ontology terms associated with each of the mouse genes. And then everybody would do their best. We'd turn them in, and they would then go and evaluate on the percent that they withheld whether or not our predictions were reasonable or not. And in fact, the contest worked fairly well. So the 9 laboratories all turned in their predictions. In general, the predictions weren't so horrible and were actually reasonable in many cases. We could make a consensus set of predictions from all 9 laboratories. So this is a diverse range of machine learning techniques, direct classifiers that people are learning, just learning gene GO-term associations. We did a hybrid approach in which we both learned, we used ensembles of classifiers to learn the associations, but we also built these linkage networks like I've described, and then integrated the information coming out of both of those. And so the consequence is that we now have a network that spans about 14,000 mouse genes. And out of the contest, the sort of synthesis of 9 laboratories' data has actually extended the annotation of the mouse gene set by quite a lot. So they're predicted annotations, but nonetheless they include something like 5,000 uncharacterized genes. They now have some new annotations for. So with these sets, we're now trying to do these same kinds of tests that we've done in the lower systems. And these, of course, we're getting progressively harder to do the follow-up experiments, the sanity checks to make sure that these are working. But what we can do at least is we can scan through the sets of known diseases for which we have genes associated with diseases, and we can see whether they appear predictive in a cross-validated sense in the networks. And we do find many genes that are predictive in a cross-validated sense. And one of these has got us particularly exciting, and that's actually one of the most common forms of human birth defects, spina bifida. So this is a massively polygenic disease involving hundreds of genes, and it actually affects something like 1 in 1,000 births worldwide, where the child is born with an exposed spine or a brain, and obviously it's a very debilitating disease. It appears from what we're seeing that the genes that are known, relatively few genes are known in humans, but in mouse models, the genes that give rise to neural tube defects cluster reasonably well in the networks. There's definitely foci in the networks that are related. So for example, all of these red genes in this sort of local area of a graph are known neural tube associated genes in transgenic mice, and all these white ones would be new candidates sitting in that region that are likely to be associated. All right, so now how do we really see whether or not those are associated? So what we've done is we've set up on a program with one of my collaborators, John Wallingford, who's a xenopus biologist, so frogs. In frogs, because they lay their eggs externally in a dish of water, you can actually go in and you can monitor the development of the embryo under a microscope. And this is actually salamander, but it gives you it's a little clearer, but it gives you the same idea. Here's a video that John had taken. What we're looking for is early in development, the developing embryo starts off as a sheet of cells and it curves up into a tube, just like you see here. And if that tube doesn't seal properly, if you go all the way through development, that's spina bifida. That causes the neural tube defect. And so if it fails to seal a little bit there, then in the embryo, then in the final animal, that's going to be an exposed spine. All right. So we can actually go in and knock out a gene early on in this process, and within essentially a day, we can see whether or not the neural tube fails to close. And so it gets us into a very testable arena. And so here's an example where this has been done experimentally, where a gene has been knocked out, and you can see that it fails to close at this stage. And so this would be a gene that now gave rise to a neural tube defect ultimately. So what we're doing is taking our computational predictions, screening in this way to find candidate genes, and then we have a pipeline set up with clinicians that have collected biopsies from human patients, where we can now start screening for genetic variation, polymorphisms, in this set of genes that we've defined in this manner. So we're sort of defining the gene set and we can now go in and do sort of targeted search for polymorphisms, because our expectation is that there's going to be a lot of small polymorphisms across the set of genes that give rise to neural tube defects in this kind of severe manner, and that we can discover them in this way. Okay, so let me go ahead and wrap up then. So I guess one of the things that I find most fascinating about biology over the last few years, and that you've been seeing through many of the talks so far in the conference, is that I'm struck by how increasingly biology is actually becoming a predictive science, and I find this very encouraging and very exciting, and it I think bodes very well for for our ability to design in medicine and these sort of things. And we've been trying to work under that notion that we can help push it towards being increasingly predictive. So I've introduced to you these notions of associated gene association networks, which we've been using as this general framework for studying gene function, and how we can reconstruct them from very diverse kinds of observations. They give a sort of a high-level description of association between genes, which turns out to have power in this guilt by association sense. And I've sort of spent more time, less on the statistics and more on the just trying to convey that that we've now been able to validate explicitly the predictions coming out of these networks now in a number of systems, yeast, worms, Arabidopsis, and moving into human cells are actually working. So I'll just wrap up again with the people that are involved, and in particular I want to point out Insuk Lee, who's helped develop much of the network work, Chris McGarry, who did all of the testing of yeast phenotypes, Ziwa Lee, who has done the experimental work that I pointed out about the ribosome biogenesis, Andy Fraser and Ben Lehner, our collaborators on the worm work, and our collaborators on the plant, Sue and Amaru. And John Wallingford in the last bit at the end. So I'll stop there. So thank you.
So you must be interested to know what source data sets you think are the most powerful. You must have an internal opinion.
Yeah, I mean, some are clearly more powerful than others.
Right.
So,
And is there any theme? Is there any theme across the whole set of species that you can extract? There was one slide you showed the precision recall. Sort of
yeah, for the individual data types.
and
Yeah.
I was sort of looking a little bit
Yeah.
about what that was. So, for you, are there some very clear data sets which are maximally informative on a genome scale? Or?
Yeah, so DNA microarrays are clearly one of the ones that inform in every experiment about all genes.
Okay.
And so they're intrinsically genome scale, and it doesn't take very many, 1,000, 500, something like that, before you, perturbing different systems, before you get at least weak inferences about lots of genes' participation in systems. So they're clearly among the most powerful ones for getting going in that. Some of them, so these complex reconstructions turn out to be another fairly powerful data set where you purify a protein, tap tag kinds of. So this is
the yeast-2 hybrid looks way out.
So yeast-2 hybrid is actually, I mean, it's not bad. It gives very specific information. What isn't so obvious from those plots is, though, the recall may be shoved way over for one of them, but those may be genes that are completely lost by many other ones. So you do need these combination of orthogonal techniques, and that's actually, I think, the biggest thing, is that no one does it all. You need the combinations of orthogonal pieces of information to get it together. But some are clearly very powerful, and so far the tap tag and the mass spec ones. I mean, the comparative genomics comes for free, so you get that no matter what, and so that's an easy one to throw into the mix that gets some other systems. So
I think you used all these different kinds of data sets. I think you left, am I correct? You didn't use phenotypic data.
we did not use phenotypic data for any of these.
And
Yeah,
you used,
that's right. So we're testing against the phenotypic data.
then you
So,
could see
yeah.
how well you could predict
Exactly,
it. But what if
yeah.
the phenotypic data is quite powerful data?
Yeah,
Now that you know
You
that,
could fold it in, yeah, and improve in some way.
Fold it in, it should make your models much better.
Yeah, you would think so. I mean, at this point, we've left them out so that we could withhold them for testing. But we could certainly go back and refold them in, and we haven't done that in most cases. So, yeah, Aaron.
Do you have an estimate of the genes that fail to validate in these types of single gene experiments? How many are actually functioning in the process,
Yeah.
but let's say have redundancy with other genes?
Yeah, so we don't have an estimate, but we do have examples where they fail to validate in a single gene experiment, but if we knock out 2, we see a synthetic phenotype that's appropriate for what we're looking at. So we actually think, based upon what the linkages are in the network and based upon those kind of experiments, that a large fraction of those is probably involved. So in yeast, we can go and look at all the pairs in a fairly straightforward way. In worm, we can do that as well in the other systems. It's not so obvious. But yeah, that's there's clearly those kinds of dependencies.
How stable, data keeps accumulating.
Yeah.
How stable is your description of the network, say the simplest one?
Yeah, so it's the most confident linkages are quite stable, and it's actually been they've been reasonably stable. We've looked at a few sort of versions as we've incorporated additional data, and they've been reasonably stable, but because we're doing it in this sort of probabilistic framework, then it's we do all of the predictions, sort of keeping the edge weights and so on throughout as we're going through the predictions. It's been reasonably stable for these updates.
Takes very strong evidence to change something.
Yeah, yeah. Except that you can often. So what we see is sort of weak edges becoming strong in later ones. We don't see strong. We never lose strong edges, but we gain weak convert to strong in later networks. So.
So in human genetics, there's a period when it wasn't possible to look at all the whole genome, and so
Right.
there was a candidate gene approaches.
Yeah, all right, yeah.
And people made these candidate lists based on all sorts of things,
Yeah.
co-publication.
Whatever, right, yeah.
And in some sense, is it fair to say that this is sort of formalizing the candidate gene
Yeah, absolutely.
type approach?
Yeah.
And maybe improving on it because you can correctly weight or co-weight
Right.
things.
Yeah.
But
Also many non-obvious aspects that only come from the functional genomics data.
right,
So,
people
yeah.
maybe underuse
Right,
the functional genomics data in
Right.
that. Though I think some people were trying to get something
Right.
out of that. But second question then is, now that we can do whole genome association studies for
So-
the sort of the story in the
Yeah.
street is, well, it turns out none of the things that have been found would have been candidates.
Yeah.
And
By existing methods, yeah.
By existing methods. So
Well-
there's sort of two issues there. One is these are often weak effects, very weak effects.
Yeah.
And maybe things with weak effects are less
Yeah.
predictable than things with strong effects. Most of the things you're looking at are knockouts.
Yeah, but actually I can address that. We've now been investigating the compatibility of these with genetic associations from genome-wide association. We can do that very easily in certain contexts. So for in yeast, there's been crosses made of strains from Rachel Brim and Lena Krugliak, where we have, for example, microarray data for 100 strains, and we know genotypes for them, and we can look at trait genotype associations. And what we see is that there's actually a compatibility. The edges that we see are enriched for trait SNP associations, and we can actually do some prioritization of candidates coming from the sort of association study using the network. So we've been exploring things like if you have a linkage interval, for example, prioritizing genes within that interval using the connectivity in the network, these sort of notions. And so it looks like that there should be some very nice ways to merge this with the genome-wide association data that could be kind of synergistic in some way. Yeah, and maybe in the back, I think yeah, Martha, go ahead.
So protein coding genes are represented by lots of different kinds of data types.
Right.
But say microRNAs,
Yeah.
you'll be lucky to, I guess, soon or
Yeah.
now start getting some kind of expression data for them.
Right.
But you're not going to be collecting all these different kinds of data types. And so I'm wondering, are those going to be dinged quite heavily because they really don't have all the different kinds of
Well-
data types feeding into your inferences?
I don't think so. As I said, microarray data is some of the most powerful data that we have. And so what's happening now is that from either the tiling array data sets or from targeted ones where people have added microarrays to the protein coding genes on an array, and then they're profiling both at the same time, we should be able to start adding microarrays as additional vertices in the graph effectively. So there's a fairly natural framework to integrate them in, and we're really just waiting for the data at this point so we can start building them into the models. Also, there's been work looking at synthetic interactions and other sorts of things involving microarrays. So there's starting to be interaction data directly targeting the microRNAs. So, yeah. Mm-hmm.
Actually, my question was related to her question as well. But one of the main data that you use in your run paper was the co-expression of the genes, if I'm not mistaken.
That's right. Yeah.
With the microRNAs, I would say you may see co-expression, while you are much more likely to see explicit expression.
Right.
And I think that will be the case for any gene that is regulated in a negative way, is like
Right.
transcriptional repression of a gene X.
Right.
In those situations, do
Yeah, you could go searching for that sort of a.
integrate another kind of data and compensate?
Well, I mean, you could go searching for those sorts of trends. Really, any particular analysis you choose to do on the relationships among the genes, it doesn't have to be correlation, that you can track in some way. I mean, the catch with the microRNAs, it'll be hard to. It's not obvious how to benchmark them, how to benchmark the associations. Since right now we're doing it all with gene ontology kinds of annotations as our sort of bronze standard we're comparing everything to, and they often fall outside of that ontology. And so I think will be a lot of technical issues in how to look at them. But intrinsically, you don't have to look at correlation. You could look at any pattern that you choose to, nonlinear, etc.
And one other question: can you ever compare the yeast networks or neural networks or human networks in terms of the networks of the interaction? Are they how much
Yeah.
correlation do you see?
Yeah, so we can't do that. The worm network includes inferences from the yeast network, and the human network includes inferences from both. So in all cases, we're data strapped. And so we're always propagating that data forward to make better models for the more complex organisms. So I agree, it would be interesting to look at those sorts of things, but in the framework that we've done this, we can't make those sorts of comparisons because they're related in some way. We can look at, that's not entirely true. We can omit all the yeast-derived linkages from the worm one, and we can look at properties of the networks in that case. And we've looked at very gross properties, but precise network structure is sort of harder to look at. But for example, there's interesting topological properties that the genes that are essential for the life of the organism tend to be the most highly connected genes in the network. They have the sum of the edge weights tends to be the highest, and that trend holds both in yeast and worm and in mouse. Even when you remove out the lower organisms. So,
Do you have any idea about your false negative rate, or will it be
oh, it's huge. Yeah.
About the total number of edges that?
Yeah, I have no idea. We think the false negative rate is quite high, and we've sort of tried to. It's a consequence of the data we have, and we've basically tried in general just to make sure we're getting a reasonable estimate of the probabilities, and in general when we're doing inferences to focus on the most confident kinds of things. So it's I'm sure the false negative rate is through the roof, but.
Any estimate of the total number of interactions?
I know the recall for some of our benchmark sets, and they're not what I would consider great recalls. So, but I think it's quite high. Total number of interactions is hard to estimate because these aren't physical interactions. These are really functional associations, which encompass both physical and logical and so on. And so it's not an obvious one how to estimate what number you should expect. Ultimately, we'd have a full adjacency matrix with everything connected to everything else with some probability. So all entries in our matrix would be filled, but only a fraction would be strong. But so there's not an obvious way to answer that. Yeah.
So going back to the question of comparing these networks with each other, I noticed that as you go from yeast to worm to mouse, the network gets denser. Is that something that explained by the level of the increasing complexity of these mechanisms, or is it just because of the fact that
You're talking about the layouts themselves, or you're talking about the sort of the number of edges per vertex? I don't think that there's an intrinsic. It's a good question. I'm not sure. I don't think that there's an intrinsic reason why that would be the case. We have more data going into each of the networks, but of course the number of genes is scaling. But it stops. When yeast to worm is the big scaling, and by the time you go worm to human and mouse, then those are all roughly the same number of genes, but we have more data being folded in at each one. What we haven't been doing is sort of folding in data from higher organisms to lower, which we could easily do, and it was just sort of historical reasons that we did it in the way that we did. So it may just be an artifact of that. So, yeah. Okay. Thank you very much. Yeah.
And,